Hello everyone, welcome to another video of Biogram. In this video, I am going to talk about pest resistance in transgenic plants. Nematodes are the very common parasites of animals as well as of plants. For example, Meloidegyne incognita. This is a nematode which infects roots of tobacco plant. They feed on the root cells and cause roots to grow large, gall or knot. That's why this nematode is known as root knot nematodes. To curb this problem, to check this problem, that the concept of biotechnology has been used, and the concept is based on the process of RNA interference. So here question arises, what is RNA interference? It is a process by which RNA molecule inhibits expression of gene. How? By silencing messenger RNA. So this is called RNA interference. RNA interference was first discovered by Andrew Fire and Craig Milo, and for their contribution in 1998 they were awarded with Nobel Prize. According to central dogma of molecular biology, DNA, it transcribes to messenger RNA, and messenger RNA, it translates into protein. In the process of RNA interference, the protein is not synthesized. And here, why it is not synthesized? Because instead of this single-stranded RNA, double-stranded RNA is formed. So when the double-stranded RNA is formed, the protein will be not synthesized. And protein is very important for the biophysiological processes which are going inside our body. In the absence of protein, the organism will not survive. This RNA interference, this process is the natural cellular defense mechanism of the eukaryotes, and it helps the organism to protect against the viral protein. So this concept is used to develop transgenic plants to combat the problem of nematodes. In this process, so to make the transgenic plant, the specific gene from the nematode is isolated and then it is introduced into the plant cell, and the introduction of that DNA is such that it synthesizes both sense and antisense RNA, and this is how double-stranded RNA is produced. When double-stranded RNA will be produced, the protein will not synthesize. So now we will understand this process with the help of diagram. First, that the specific gene isolated from the first the specific gene was isolated from the nematode and then it is introduced into the plant cell. So since the nematode is infecting the root of the plant, so here I am drawing one root cell. So this is the root cell, and in the root cell the specific gene has been introduced. So this is double-stranded DNA, and this double-stranded DNA, it will transcribe in such a way that both the strand, that is antisense and sense, both will transcribe into RNA. So here so this is DNA, RNA, here the RNA is synthesized. For RNA, to represent RNA, I am using black color here. So these two strands of the RNA, they are complementary to each other. So this is how that the double-stranded RNA is formed. So this is double-stranded RNA, here I will write DNA, and this is double-stranded RNA. Here one enzyme is there which is present in the cell, that is RNAs, and this is also called, that is dicer. The dicer enzyme, it will cleave the double-stranded RNA into small fragments, and so these are the small fragments of double-stranded RNA. These fragments are called small interfering RNA. These small interfering RNA, they will form a complex with an enzyme that is called ribonucleases. So with the help of ribonucleases, it will form a complex, and that complex is called RISC. RISC stands for RNA-induced silencing complex. When a nematode comes in the contact of the plant cell, that is the root cell, then the small interfering RNA, they enters the cell of the parasite. So here they will enter. So here, that is, I have used black color. So this, they will enter here. And here, since it is a eukaryote, here also the same process, that is the transcription and translation, that the process is going on. So here in case of nematodes, that is the DNA will transcribe into messenger RNA, that is the single-stranded RNA. So here that this complex, that the RISC, that RISC, it will identify the messenger RNA. This here, this will identify messenger RNA, and it will cleave to small pieces, and the unzipping of small interfering RNA will take place, and this, so it will become single-stranded, and this unzipped small interfering RNA, it will bind with the RNA molecule, and this is here, and hence that the double-stranded RNA will form, and the messenger RNA will be destroyed. So like this, that as I told you, that the RISC RISC, then it will break the messenger RNA into small fragments, and the small interfering RNA, it will bind and destroy the messenger RNA. Once the messenger RNA will be destroyed, the translation will not occur, and so the protein will be not synthesized. And in the absence of the protein, eventually the parasite will die. This is how the transgenic plant will protect itself against the infection of the nematodes. Flower saver tomato is transgenic tomato. Usually country tomatoes, they quickly get softened due to the presence of enzyme, that is polyglacturonase. And these tomatoes which are transgenic, in these plants, that the gene which is responsible for the synthesis of this enzyme has been removed. So these tomatoes can be preserved for a longer period of time, and their flavor and texture can be intact for a longer period of time. So this was one example. Other example we can take, that is golden rice. Golden rice is transgenic variety of rice, and which is rich in vitamin A. So here, to make the transgenic variety of rice, that the gene which is responsible for the synthesis of beta carotene, it is isolated from the daffodils and it is introduced into the rice plant. And there that the gene which is responsible for the beta carotene, it makes the transgenic rice rich in beta carotene, which is synthesizes vitamin A. So this was all about the different types of transgenic plants. You must have observed that due to genetic modification, that the genetically modified crops are useful in many ways. First of all, they are resistant to biotic factor and abiotic factors too. Third, that the nutritional value of the food is quite good. Genetically modified crop, they clean up heavy metal pollution from the contaminated soil. So this was all about the application of biotechnology in agriculture. In the next video, I will discuss about application of biotechnology in medicine.